In Lab 4, we're going to be looking at the enumeration of microorganisms, ways in which we can determine the number of microbes such as bacteria in a sample. There are a number of videos for you to watch for this one, as well as, again, a pre-lab quiz. Most of these videos deal with techniques we'll be using in lab, how to read and aseptically handle a sterile 1 milliliter pipette, how to read and aseptically handle a 10 milliliter pipette, how to pipette using aseptic technique and use a vortex mixer, and how to obtain single colonies for counting using the splint plate method. Now this lab involves a little bit of arithmetic. Don't even want to hardly say math, so some people look at this and say, oh, it looks like math, I guess I won't understand it. But it's simply adding, dividing, and multiplying with a calculator. That's as complex as it gets mathematically. So again, anyone can do these problems if you follow how to do them step by step. So again, it's often important to be able to determine the number of bacteria in a given sample. But remember, in dealing with microorganisms, we're dealing with huge numbers, very large numbers. So often the work is done using scientific notation. But again, in this lab and on the problems on the quizzes, you can use either scientific notation or old longhand math with all the zeros, as long as you can get the right answer. Now, before we get started on the ways we can determine the amount of bacteria in a sample, we need to look at Appendix B. If you remember, I told you that you need to read Appendix B and Lab 4 before you come to class to be prepared so that we can get this lab finished in the time allotted. So let's take a look at Appendix B in your lab manual. Now, the first part of Appendix B just deals with scientific notation. It reviews how to write numbers in scientific notation if you wish to use them on your quiz, or if you want to review how to do that, or if you want to learn how to do scientific notation. Remember, in scientific notation, there's only one digit, a number between 1 and 9, to the left of the decimal point, and the rest of the number is expressed as an exponent. So I'm not going to go through that here because that was one of your prerequisites coming in. But again, if you're really uncomfortable with scientific notation, you can do the problems longhand. What I do want to go over, though, is part 2 of Appendix B on dilutions. It's essential you know what the two numbers in a dilution mean and what the term dilution factor means. So looking at a very simple dilution, if we put 1 milliliter of bacteria in 1 milliliter of sterile saline, the total number of milliliters in that tube is now 2. So you see the math isn't too hard. 1 plus 1 equals 2. And of that 2 milliliters, 1 milliliter is bacteria. So we call that a 1:2 dilution. It's also written as 1/2. And that tells us that this tube now has half as many bacteria as in the original milliliter because we've just doubled the volume, but we have the same number of bacteria. So remember, the lower number in your dilution fraction, or the second number in your dilution, is the sum of all the milliliters in the tube in this case. Another example, 1 milliliter of bacteria is mixed with 3 milliliters of sterile saline. So there's now 4 milliliters in the tube, 3 plus 1, one of which is bacteria, so that is a 1:4 dilution. And that tube has 1/4 as many bacteria per milliliter as the original milliliter. Now, the one we use most frequently in science is 1 milliliter of bacteria is mixed with 9 mils of sterile saline. So 1 mil and 9, that means there's a total of 10 milliliters in the tube, one of which is bacteria, and that is a 1:10 dilution. And we like that because now we can also start writing the number in scientific notation. 1/10 is the same as 10 to the minus 1. Now, 1 to 10, or sometimes 1 to 100, is about as high as we go on a straight dilution. But today we need to dilute the bacteria 1 million times, 1 to 1,000,000. And it's not really feasible to pipette 1 mil of bacteria into 999,999 mils of water or saline. So what we normally do for any dilution greater than 110 is make a serial dilution, dilutions of dilutions. There's a simple example here, but let's take a look at the actual dilution we're doing in today's lab with this link. So we're going to take a sample of E. coli. We're going to take 1 ml of the E. coli and put it in 9 ml of sterile saline. So 1 ml in 9 ml is a 1 to 10 dilution. We now have 10 ml in the tube, 1 is bacteria. And of course, 1 to 10 can also be written as 10 to the minus 1, the same number in scientific notation. Now, once we add the bacteria, we mix this thoroughly on the vortex mixer so the bacteria get evenly distributed. Now, if we take 1 milliliter out of the 1 to 10 dilution tube and put into 9 ml of sterile saline, 1 ml in 9 ml is 1 to 10, but it's 1 to 10 of 1 to 10. And 1/10 of 1/10 is 1/100th or 10 to the minus 2. So a milliliter in this tube now has 1/100th the number of bacteria in the original milliliter. We mix that thoroughly on the vortex mixer to get them evenly distributed. Now we take 1 ml out of our 1 to 100 dilution and put that into 9 ml of saline. So 1 to 10 of 1 to 100 is 1 to 1,000 or 10 to the minus 3. We mix that thoroughly. Take 1 ml out of the 1 to 1,000 dilution, put that into 9 ml of saline. 1 to 10 of 1 to 1,000 is 1 to 10,000 or 10 to the minus 4 in scientific notation. We mix that thoroughly. Take 1 ml out of our 1 to 10,000 dilution, put into 9 ml of sterile saline. That's 1 to 10 of 1 to 10,000, which would be 1 to 100,000 or 10 to the minus 5. And if we do that one more time, that would give us 1 to 1,000,000 or 10 to the minus 6. So as you see, each successive tube has 1/10 as many bacteria per milliliter as the previous tube. And now we know that 1 ml from this tube, the 10 to the minus 4 tube, has 1/10,000th the number of bacteria in the original mill. We know that 1 milliliter in our 10 to the minus 5 tube has 1/100,000th the number of bacteria in the original mill. And 1 milliliter of bacteria in our final tube has 1/1,000,000th the number of bacteria of the original. So once we get them diluted enough, we're going to be able to plate them out in lab, as we'll see shortly, count the colonies, and by knowing the colonies and the dilution of the tube, we can extrapolate how many bacteria were the original sample using simple formulas. So that's making dilutions. And the dilutions, of course, are all fractions of the milliliter, so they're all negative exponents in scientific notation. But when it comes time to calculate the total of number of bacteria in that original sample, we have to use the term dilution factor, not dilution. And dilution factor is simply the inverse of the dilution. And in arithmetic, inverse simply means you flip the two numbers of a fraction over. That's the inverse. If you're writing it in scientific notation, the inverse of a negative exponent will be a positive exponent. So if we go back to our samples up above, if the dilution was 1 to 2, the dilution factor is the inverse of that, 2 over 1 or 2. And that makes sense because this tube has half as many bacteria as the original sample, so the original sample must be 2 times that. If we had a dilution of 1 to 4, then the dilution factor is the inverse, 4 over 1. That's 1/4 the real number, so the real number must be 4 times that. If the dilution is 1 to 10 or 10 to the minus 1, then the dilution factor is the inverse, 10 over 1 or 10 to the positive 1 in scientific notation. That was 1/10 the number of bacteria originally, so the real number must be 10 times that. And if we go all the way up to our last dilution tube we looked at, if the dilution is 1 to 1,000,000 or 10 to the minus 6, the dilution factor would be the inverse, 1,000,000 over 1, or 1,000,000, or 10 to the 6th in scientific notation. So the dilution factor is a whole number we have to multiply the dilution by to get the original number. So with that look at dilutions and what the numbers of dilutions mean and what dilution factors mean, let's return back to lab 4 and go over the content. Now I'm going to approach this kind of backwards. We'll do the plate count last because that's what we're actually doing in lab today. So let's move down to part C of our lab on turbidity. Now we're not going to do a lab on turbidity or absorbance, but I want to explain the concept to you behind that, because that's kind of important to at least know the concept. And there's a couple of objectives on the content here, but that's about it. There won't be any problems on this, but there's a few terms we have to know. So we saw in lab 2 that when bacteria grow in a liquid medium, the culture typically appears turbid or cloudy because the bacteria block and reflect the light passing through the tube. So the light the bacteria absorb is directly proportional to the concentration of bacteria in the culture, the number of cells in the culture. The greater the number of bacteria, the more light they absorb or block. And we can then use absorbance as a way to eventually get a number of bacteria by combining absorbance with one of the other techniques we're using today. Now, again, we're not going to do that because it takes too long. If you had BIOL 110 at Catonsville, you did a couple of experiments using a spectrophotometer and making a standard curve, and it works the same way when we deal with determining bacteria. So the machine we use to measure turbidity or absorbance is a spectrophotometer, which looks like this. The tube of bacteria goes in this well, a light goes through the tube and hits a photometer, and that gives you a reading up here in very simple terms. So there's two ways we can describe the ability of the culture of bacteria to block light. We can express it either as percent of light transmitted through the tube or the amount of light absorbed in the tube. And if we look at figure 4, we can see what that means. So when the light goes through the tube, we can either measure the amount of bacteria, the amount of light that the bacteria block in the tube, the amount of light that doesn't get through, the amount they absorb. That's called absorbance. And the more bacteria in the tube, the greater the absorbance. The greater the number of bacteria, the more light they absorb. But we can also measure the percentage of light transmitted through the tube, the amount of the light not blocked. And that's inversely proportional. The greater the number of bacteria in the tube, the less light gets through the tube. So the smaller the percent light transmitted. And so one of your objectives asks you to explain those two relationships. That the percent of light transmitted is inversely proportional to the bacterial concentration. In other words, the greater the number of bacteria in the tube, the lower the percent light transmitted, the less light gets through the tube. Or if we measure absorbance, also called optical density, absorbance is directly proportional to the concentration of bacteria. The greater the number of bacteria, the greater the absorbance. And normally we measure absorbance because we can plot that out on regular graph paper if we were doing a standard curve. So what's often done in research and industry and such is they combine the spectrophotometer, the absorbance, with one of the other counting methods like the Petroff-Hauser counter or the plate count that we'll be talking about shortly. And the way that works in general is that you'd make several dilutions of the bacteria, and then you would count the number of bacteria in each different dilution tube using either the direct microscopic method or the plate count method. And then you'd use a spectrophotometer to measure the absorbance of each dilution tube. And once you have these two variables, the number of bacteria and the absorbance, you can prepare a standard curve that compares absorbance to the number of bacteria made by plotting absorbance versus the number of bacteria. And that's directly proportional, as we said. So although there's no numbers on the side of this in terms of the number or absorbance, that would eventually have numbers written there, and this would be the absorbance measured. But if we plotted out several different dilutions, we would get a standard curve that's directly proportional, because we know the greater the number of bacteria, the greater the absorbance. And so once we've made a standard curve, then we could take any dilution tube of that organism under the same conditions, put that in a spectrophotometer, find the absorbance, and then from the standard curve, find out what number that represents. So we could take any absorbance of any dilution of that culture, find the absorbance, go over the standard curve, go down and see what number of bacteria that corresponds to. So by combining the technique, we can actually get numbers using absorbance by preparing standard curves. But again, that takes about 3 hours to set up and do, and we don't have time to do that. So we're just looking at the concept that absorbance can be used indirectly as a measure of growth. The greater the number of bacteria, the greater the absorbance. So again, just a couple of objectives that deal with terminology for turbidity, but there's no problems we have to do on that. Now, you will have to be able to do a problem regarding the direct microscopic method or total cell count. And this is a technique that gives us an actual count of the number of bacteria using a microscope. So this uses a special counting chamber. It's a highly precise microscope slide that has ruled lines on it and a cover slip that sets up a specific height. And so when you look through the microscope, the ruled lines create little squares that have a known volume in them. So the concept behind the direct microscopic method is that the number of bacteria in a small known volume is directly counted microscopically. We have to get down to a very tiny number of bacteria to actually count them. Remember, 1 milliliter of bacteria could contain up to 1 billion bacteria. We don't want to count 1 billion. So we count a very tiny volume that has a feasible number to count. And then once we know how much is in the smaller volume, from there we can extrapolate to how much is in the larger volume. So the special counting chamber is called a Petroff-Hauser counting chamber. And these are quite expensive, something like $600 a piece. So we're not going to do the actual count, but you should be able to do a hypothetical problem. Now, the way this counting chamber is made up is that if we look at figure 2A here, etched in the glass are these big double-line squares. Now, there's a whole bunch of these double-line squares on the slide. This is just one of them. That's what we're counting, the big double-line square. This is divided into 16 smaller squares to make counting easier. But this is the volume we're counting. So the way the slide's made is that this square holds a volume of 1/1,250,000th of a cubic centimeter. And since we're basically dealing with the bacteria diluted in saline or water, a cubic centimeter is the same as a milliliter. So we're counting the number of bacteria at a very tiny known volume, 1/1,250,000th of a cc. But then technically, if we invert that number, this is the dilution, remember, of the cc. If we invert that, then this number times 1,250,000 would roughly represent the number of bacteria in the original cc. So there's a formula we use for this. And we're trying to determine then the number of bacteria per cubic centimeter in the original sample of E. coli. And when you have the quiz problems, you'll have the formula. I'll provide that, and I'll provide a calculator. So the average number of bacteria per cubic centimeter equals the average number of bacteria per large double-line square. Now, the number in a double-line square we get by actually counting. And here's what the Petroff-Hauser counter actually looks like under a microscope. So this is one of the large double-line squares. There's a whole bunch of these on here. And these are the actual bacteria you see. So if we count a square, we're counting the number of bacteria in this volume right here. And that volume, again, is 1/1,250,000th of a cubic centimeter. But as you'll see from the formula, it says the average number per large double-line square. So to make it a little more accurate, we don't count just one of these volumes here, one of these double-line squares. We typically count maybe 5 of them. and then take an average. So we count this square, then we move over and count another big square, move it over, count another big square, until we do 5 of them. So that's why it says the average number of bacteria per large double-lined square. Now, since you won't be counting, we'll give you the numbers, and I'll show you how in the practice problems coming up shortly. Now, we multiply the average number of bacteria per large double-lined square by the dilution factor of the large square. Now, remember that large square we just said holds 1/1,250,000th of a cubic centimeter. That is the dilution of the cubic centimeter, 1 over 1,250,000. So if we invert that, remember dilution factor we just learned is the inverse of the dilution. We have to flip the fraction over. So the dilution factor of the square will always be the same number. It's a constant for that slide, 1,250,000. Or in scientific notation, that would be 1.25 times 10 to the 6. And then finally, we multiply that by the dilution factor of any dilutions we made before we put the sample of bacteria in the counting chamber. And what we normally do is mix the bacteria with dye first. Now, the reason for that, as we will be seeing in our staining labs, is that bacteria, of course, are very tiny. They're very hard to see if they're not stained. So by mixing the bacteria with dye, we can see them more clearly. Another problem, if they're living bacteria we put on the slide and they're motile, they could be swimming from square to square. That'll make counting quite difficult. So the dyes also kill the bacteria. So by mixing the bacteria with dye first, that makes them easier to see on the slide. They become stained, and the dye kills them, so they stop swimming. But remember, we're adding dye. That also dilutes them. We have to take that into account. So that is our formula. But there are 2 practice problems you can do now to see how to do one of these problems. And we'll take a look at those. So for the direct microscopic method to do your practice problems and see how to do this on a quiz, scroll down on your online lab manual to the performance objectives. And you'll notice that performance objective number 5 under discussion says, when given the total number of bacteria counted in a Petroff-Hauser chamber, the total number of large squares that are counted, and the dilution of the bacteria placed in the chamber, calculate the total number of bacteria per cubic centimeter in the original sample. And here's your 2 practice problems. So let's look at the first one. This tells you that 1 milliliter of E. coli is mixed with 3 mils of dye. Now again, that will stain the E. coli so we can see them better under the microscope. It'll kill them so they don't swim from square to square, but it also dilutes them. Well, that's the 3rd factor in our formula, the dilution factor of any dilutions we made before we put it on the slide. So again, since we won't have time to actually count during a lab quiz, I'll give you the counts that were obtained. And of course, remember the square always holds the same thing. The way the square is made, it holds 1/1,250,000th of a cubic centimeter. So let's say we mixed 1 mil of E. coli, we put that in the counter, and we count 5 squares, and we get these numbers. So let's say the first square had 46 bacteria. We move the slide over. The second square had 50. The third one had 54. The fourth square had 58. The final one had 52. So we counted 5 squares. From that and the formula, you should be able to plug in the numbers and find out how many bacteria per cc in the original sample. So if you're not sure how to do that, now let's click on the answer page and it takes you through this step by step. So again, here's the formula. We're trying to find the number of bacteria per cc. That's equal to the average number of bacteria per large double-lined square. That's where we counted 5 squares and we're taking an average. So again, it's addition and division with a calculator. We multiply that by the dilution factor of the large square. Well, again, that's a standard. Remember, the large square holds 1 over 1,250,000th of a cc. So if we flip that over and invert it, the dilution factor of the square will always be 1,250,000. That's the way the square is made. If you're doing it in scientific notation, that would be 1.25 times 10 to the 6. Then we multiply that by the dilution factor of any dilutions we made before we put the sample on the counting chamber, such as mixing it with dye, which we did. So going to our formula, first part says we have to find the average number of bacteria per large double-line square. So the problem gave you our counts. These were the counts on the previous page. For the 5 squares, we add them up. That's 260. Now we counted 5 squares, so we divide 260 by 5 to get an average. So the average would be 52, 260 divided by 5. So that's the first number in your formula, 52 if you're doing it longhand. If you do it in scientific notation, then 52, remember, is 5.2 times 10 to the 1 in scientific notation. Then we multiply the average number of bacteria per large square, 52, which we just found, by the dilution factor of the square, which is always 1,250,000 or 1.25 times 10 to the 6 in scientific notation. Again, that's because the square holds this dilution of a cc. So if we invert that, then it becomes 1,250,000 over 1. So 52 times 1,250,000, if you use your calculator, is 65 million. If you're doing the problem in scientific notation, then it would be 5.2 times 10 to the 1, the average number of bacteria per square, times the dilution factor of the square, which is 1.25 times 10 to the 6 in scientific notation. You give us a final answer of 6.5 times 10 to the 7th. So that's this number in scientific notation. Then finally, the third part of the formula, we multiply the dilution factor of any dilutions made before we put the bacteria in the counting chamber, such as mixing with dye. Well, in this case, we told you that 1 ml of bacteria was mixed with 3 ml of dye. Remember, we have to add them up to get the bottom number, so that's a 1:4 dilution. So the dilution factor is the inverse, 4 over 1 or 4 when we flip it over. So going back then, we have the 65,000 from the previous step times 4, giving us a final answer of 260 million E. coli per cubic centimeter. If we're doing it in scientific notation, then the 65 million is 6.5 times 10 to the 7th times 4, giving us a final answer of 2.6 times 10 to the 8th if you're doing it in scientific notation. So that's the first practice problem. And let's look at the second one for the direct count now. In this case, 1 ml of E. coli is mixed with 1 ml of dye. Remember, that's the third part of the formula. So in this case, it would be 1 ml of bacteria, 1 ml of dye is 1 to 2. We have 2 mls, 1 is bacteria. The slide always holds the same volume, of course, but here's the counts we have for this one. The first square had 61, second one had 74, third had 78, fourth square had 63, final had 64. So again, if we take that and plug it into our formula, the first thing we have to do is find the average number of bacteria per large double-line square. So here's the 5 squares we counted. We add those up. That's a total of 340. To find the average, we divide 340 by 5, because we counted 5 squares. And that means there's 68 bacteria per square. Or in scientific notation, 6.8 times 10 to the 1 bacteria per square. We multiply that by the dilution factor of the square. There's a typo there. I will correct that right after we're done there. That will be 1,250,000. Again, that's a standard for the slide. So 68 times 1,250,000 is 85 million. In scientific notation, then, 68 would be 6.8 times 10 to the 1. 1,250,000 would be 1.25 times 10 to the 6. So the final number there would be 8.5 times 10 to the 7th. So that's the second part of the formula, multiplying the average number per square by the standard dilution factor of the square, which is always 1,250,000. And then finally, we multiply that by the dilution factor of any dilutions we made, such as mixing with dye, before we put it on the slide. Since in this case we mixed 1 ml of bacteria with 1 ml of dye, that's a 1:2 dilution. We have 2 mLs in the tube, 1 is bacteria, and the inverse of that is 2. So 85 million from step 2 times 2 gives us a final answer of 170 million bacteria per cc. If you're doing a scientific notation, then it's 8.5 times 10 to the 7th times 2 is 1.7 times 10 to the 8th. And I'll make that correction up here so that when you see it online, it'll be the right number there. So those are 2 practice problems you can do to get ready for the direct microscopic problem that'll be on Lab Quiz 1. And remember, Lab Quiz 1 covers the introduction to the lab manual and labs 1 through 4. So it'll go through up and include this lab. And that's the direct microscopic method. Now, the method we're actually doing in lab today is called the plate count or viable count. So again, because we have huge numbers of bacteria in a milliliter of fluid, up to 1 billion, we have to serially dilute the sample to get a feasible number to count. But in this case, we're not going to count the number of bacteria. We're going to put samples of the bacteria on an agar surface and count the number of colonies that form. So if we dilute the bacteria out far enough, and we use the spin plate method to plate them out on a Petri plate and separate the individual bacteria, we can, as we see in figure 1, count the colonies that form. And if we know how many times we diluted the bacteria to get to those number of colonies, we can find the number of bacteria in the original sample. But as we see, we're going to actually call this colony forming units in the original sample rather than bacteria. But it's called the plate count because we're counting colonies on a Petri plate rather than counting bacteria under a microscope. But the reason we're not saying the number of bacteria per milliliter is, remember, a lot of bacteria come in arrangements, multicellular arrangements. If we were counting a streptococcus, then a chain would produce a colony. We have no idea how many bacteria were in the chain. It could just be a couple, it could be a dozen. So we don't worry about it. We simply call these colony forming units. If a chain forms a colony, that's a colony forming unit. If we're counting a staphylococcus, then whatever size the grape-like cluster would be that forms a colony, that would be a colony forming unit. If we're counting a single bacillus like E. coli, then actually one bacteria would represent one colony. So what we do in this case, we're trying to determine the number of colony forming units in the original sample. The number of colonies that we count, and keeping in mind that the original colony could have come from one bacterium or maybe one arrangement of that bacterium. We simply call it a colony forming unit, whatever forms a colony. So we start out by diluting the bacteria so we get a reasonable number of colonies to count. And then after incubation, we look at the plates and we have a ground rule here. We only count a plate that has between 30 and 300 colonies. So all this work today is to get one usable Petri plate defined as one that has between 30 and 300 colonies. So the plate I showed you in figure 1 would be a good plate to count. That definitely has less than 300, but more than 30. So the formula we use for the plate count is a pretty simple one. We're trying to determine the number of colony forming units, or CFUs, per milliliter of the original sample. That's going to be equal to the number of colonies we count next time. Remembering our ground rule, we only use a plate that has 30 to 300 colonies. And we multiply the number of colonies times the dilution factor of that plate. So you simply find the dilution on the plate, invert the dilution, flip it over. That's the dilution factor. So let's say that we made this dilution. This is the dilution we're making today. So we're going to make a serial tenfold dilution here, series of 1 to 10 dilutions. And this is going to dilute it all the way to 1 to 1,000,000. And then once we dilute the bacteria, we're going to put 1/10 of a milliliter from the tube onto a Petri plate, spread that out with a turntable and bent glass rod to separate the bacteria in that 1/10 of a mil, incubate it, and then count the colonies. So let's say next time the Petri plate that we plated from this tube, that 1/10 of a mil gave rise to 22 colonies. If we take 1/10 of a mil from this tube and put it on a Petri plate, let's say pretend numbers there, this plate showed us 150 colonies. And we take 1/10 of a mil from this tube, that showed over 400 colonies. So again, we'll give you made up numbers for the quiz. Of course, in lab next time, you'll be choosing the correct plate, the one that has between 3,300, do the count and plug the numbers in and put them up on the board. So let's see how we go about determining our answer here. So again, this is the dilution we're making, and you'll have to determine what the dilutions are in the tube, but remember, each tube is 1/10 of the previous tube. So you just add another 0 to the denominator each time, or add another exponent if you do it in scientific notation. So again, if we put 1 mil of bacteria into 9 mils of saline, we have 10 mils in the tube. That's a 1 to 10 or 10 to the minus 1 dilution. If we take 1 mil out of here, put it in 9 mils of saline, that's 1 to 10 of 1 to 10, which is 1 to 100 or 10 to the minus 2 dilution in this tube. We take 1 mil out of the 1 to 100 dilution tube, put in 9 mils of saline, 1 to 10 of 1 to 100 is 1 to 1,000 or 10 to the minus 3. Take 1 mil out of there, put 9 mils of saline, gives us 1 to 10,000 or 10 to the minus 4. Take 1 mil out of the 1 to 10,000, put in 9 mils of saline, that's 1/10 of that, so that's 1 to 100,000, 10 to the minus 5. And of course, the last two would be 1 to 1,000,000 or 10 to the minus 6. But remember, we're not going to when it comes time to count, we don't have to use this plate, because remember, we can only count a plate that has 30 to 300. So that's the only plate we can count, 150 colonies. So this plate had over 400, we wouldn't use it. This had only 22, we couldn't use it. This is the correct one. And so the dilution in this tube is 1 to 1,000,000 or 10 to the minus 5. That means 1 milliliter in this tube has 1/100,000th the number in the original milliliter, because we diluted the original milliliter 100,000 times. So what we're supposed to put on the Petri plate is 1 mil, because this fraction represents the amount in 1 mil. But 1 milliliter is too much fluid to put on a standard Petri plate. You wind up having standing water on the plate. You can't spread them out. They all grow together. So 1 milliliter is too much fluid to put on the plate. So we're forced to put only 1/10 of a mil on the plate. And that's why it says on the plate, you'll notice that'll be 1/10 the dilution in the tube. And the reason the dilution on the plate is 1/10 of the tube is that only 1/10 of a milliliter was put on the plate, not 1 mil. So 150 would be 1/10 the amount that was actually in the milliliter here. And so all we have to consider when we're doing our problems is that the Petri plate will always be 1/10 of the dilution in the tube it came from. So if this tube is 10 to the minus 4 or 1 to 10,000, the dilution on this Petri plate would have been 1 to 100,000 or 10 to the minus 5. Since this tube has a dilution of 1 to 100,000 or 10 to the minus 5, 1/10 of that makes the dilution on the plate 10 to the minus 6 or 1 to 1,000,000. Now again, we're only showing this one because that's the only one you need to know. That's the one that's usable to count. But remember, 1/10 in this tube would be 1 to 1,000,000 or 10 to the minus 6. So the dilution factor on that plate would be 1 to 10 million or 10 to the minus 7. So when we get to our practice problems in a few minutes again, you have to remember to take the dilution all the way down to the plate. but it's really no harder than counting till we get to the Petri plate. If we go back to the previous page, and you saw this problem on a quiz, well, if you're trying to find out, first thing you'd have to determine which plate to count. The only one we can count is 150 because that's the only plate that has between 30 and 300 colonies, so we ignore the other two. Then we have to find what's the dilution on this plate. It's real easy in scientific notation because you just count. 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5, 10 to the minus 6 to the plate. So the dilution on the plate's 10 to the minus 6. Or if you're doing it longhand, add a 0 each time to the denominator. 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000, 1 to 1,000,000. And then when it comes time to do the problem, it'll be 150 times 1,000,000, the inverse of 1 over 1,000,000, or 150 million. So we're going to show you a couple of practice problems you can do on that now, found in your online objectives. And again, you want to practice these before the next lab because you'll be counting the colonies next time and putting your results on the board. So here's our objective 2 under discussion, gives us 2 plate count practice problems. And in this case on the quiz, I'll give you a diagram up on the board with that type of information, and you would calculate the number of colony forming units per milliliter. So as we see in example 1 here, a sample of E. coli is diluted according to the above diagram. The number of colonies that grew is indicated on the Petri plate. How many colony forming units are there per milliliter in the original sample? So again, we'll click on the answer in a second, but remember just 2 steps here. Choose the right plate, one that has 30 to 300. Find the dilution to that plate and multiply those 2 numbers. So let's look at our step-by-step answer. Here's the formula. The number of colony forming units per milliliter in the sample equals the number of colonies we count next time, remembering that we're only going to count 1 plate, one that has between 30 and 300 colonies. And we multiply that by the dilution factor of the plate. We find the dilution on the plate and we invert it to get the dilution factor. So the first thing you do is choose the correct plate to count. Has to be between 30 and 300, so the only one we can use is 63. So we're going to use this Petri plate. And in scientific notation, that would be 6.3 times 10 to the 1. Then we have to find the dilution on that plate. Well, like I said, we just count. So if we do it in scientific notation first, this would be 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5, 10 to the minus 6 to the plate. Because remember, the plate's always 1/10 of the tube because we're only putting 1/10 of a milliliter on the plate. So we know the dilution of the plate is 10 to the minus 6 in scientific notation. If you're doing it longhand, then remember it's going to be 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000, 1 to 1,000,000 on the plate, 1 over 1,000,000. So the dilution on the plate is 1 to 1,000,000 or 10 to the minus 6. And now we multiply those 2 numbers. So 63 times 1,000,000 is 63 million. That would be your answer if you're doing it longhand. 63 times 1,000,000. Remember, you had to invert the dilution. The dilution is 1 over 1,000,000, so the dilution factor is going to be the inverse, or 1,000,000. If you do it in scientific notation, then 6.3 times 10 to the 1 is the number of colonies. The dilution factor of this plate is 10 to the positive 6, so your final answer is 6.3 times 10 to the 7th. Remember, when you multiply exponents, you add them. So 6 plus 1 is 7. That's where you get the 7th. And here's the second practice problem. I'll just point it out to you and then let you actually get a chance to practice that one. Oops, hit the wrong problem. Sorry, that's the plate count. We want the plate count. There we go. So here's a dilution. Now don't just memorize the what the plates are, because remember, we could use any number of tubes. Notice this one has 5 tubes, not 6. We might use 7 tubes, how far we think we may have to dilute it. So remember, it's just a matter of choosing the correct plate, one that has 30 to 300, in this case 161. Find the dilution on that plate, 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5, 10 to the minus 6 to the plate. Or if you're doing it in longhand, 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000, 1 to a million. So 161 times a million for 161 million, or 1.61 times 10 to the 2, that's 161 in scientific notation, times 10 to the 6th, 1 million, the inverse of 10 to the minus 6, and there you'd have your answer. So again, there's step by step on how to get the answer. Go through and practice that. So that's the concept. Now, what we're doing today in lab is only the plate count procedure. And we'll be demonstrating how to pipette aseptically. Remember, I've also shown you videos on how to pipette aseptically using a 1 mil pipette, which is all we'll be using in today's lab. I've also indicated how to use a 10 mil pipette in a different video. Also, I've showed you how to use the vortex mixture, because remember, every time we add bacteria to a new tube, we have to mix them thoroughly to get the bacteria evenly distributed before we take them out to put into the next tube. And there's also a video on how to do the spin plate method. So basically, you're just going to follow this diagram for your dilution in lab. We'll put that up on the board. You're going to aseptically pipette 1 mil of E. coli into 9 mils of saline for 1 to 10 dilution. You're going to mix that thoroughly on a vortex mixer. Take 1 mil out of your 1 to 10 dilution and put into 9 mils of saline for a 1 to 100 dilution, or 10 to the minus 2. Mix that on the vortex mixer. Aseptically remove 1 mil from the 1 to 100 tube, put that into 9 mils of saline for 1 to 1,000 or 10 to the minus 3. Mix that. Take 1 mil out of the 1 to 1,000, put into the next tube for 1 to 10,000 or 10 to the minus 4. Mix. Take 1 mil out of there, put in 9 mils of saline for another 1 to 10, 1 to 100,000 or 10 to the minus 5. Mix. Take 1 mil out of there, put in 9 mils of saline for a final dilution of 1 to a million or 10 to the minus 6, and mix. So we're going to use the first 1 mil pipette to do all of these tube-to-tube dilutions till you get all the way down to the 6th tube. Then discard that pipette in the beaker we use for discarded pipettes and swabs. And you're going to take a second 1 milliliter pipette, a new one, and use that to put 1/10 of a mil of bacteria from each of the last 3 tubes onto the corresponding Petri plates. Now, we're only using the last 3 because these are probably going to be way too concentrated. There'll be well over 300 colonies on the sample. So hopefully there's a good plate to use in this range. And remember, when you label your tubes, label them just like the diagram. 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5, 10 to the minus 6. And then label your Petri plates. Remember, they're 1/10 of what's in the tube because we're putting a 1/10 of mil on, not 1 mil. So the plates will be labeled 10 to the minus 5, 10 to the minus 6, 10 to the minus 7. And once you have everything labeled, just using your pipette to follow that diagram. Now, when you get ready to put the bacteria on the Petri plate, start with the most dilute tube, because if you started with the most concentrated tube, there's always some bacteria left inside the pipette that doesn't come out thoroughly. And so there would be a greater chance of having some increasing the counts of the successive tubes that way. But if we start with most dilute, that's not going to have much effect. So you're going to aseptically remove 1/10 of a mil from your 10 to the minus 6 or 1 to 1 million tube and pipette that in the center of your 10 to the minus 7th Petri plate. Then immediately flame the glass rod, as we'll show, using alcohol. Put the plate on a turntable, spin it for about 30 seconds, and spread that 1/10 of a mil out over the entire surface of this plate to get them evenly distributed. Then take 1/10 of a mil of your 10 to the minus 5 or 1 to 100,000 dilution and pipette that in the center of this plate. Again, sterilize the glass rod. Using the turntable, spread that 1/10 of them out over the entire surface. Then finally take 1/10 of a mil out of your 1 to 10,000 or 10 to the minus 4 tube, put on your 10 to the minus 5 plate, and spread that out with the bent glass rod and turntable. And then you'll simply be incubating these 3 plates upside down in your Petri plate holder at 37. And next time we'll look for a plate that has between 30 and 300 and count the number of colonies. Now, once you've plated out these 3 plates, all your dilution tubes can go back in the hood. We're done with them, so you can dispose of them safely in the baskets. And of course, the used pipettes go in the used pipette container. So again, if you follow that diagram, that's pretty much all there is involved there. But remember, there's pictures here to remind you that in a 1 mil pipette, from the tip all the way up to the 0 is 1 milliliter. That's why it's a 1 milliliter pipette. Between any 2 numbers is 1/10 of a mil. From 0.9 to the bottom is also 1/10 of a mil. That's sometimes considered not as quite as accurate as going from number to number, but it won't make much difference in our lab if you just use this 1/10. So remember, when you're going from tube to tube, you're going to fill it all the way up to the 0, put the whole thing in. When it comes time to put the bacteria on the Petri plate from the tube, then we're using 1/10 of a mil. And we use blue fillers for 1 mil pipettes. So again, make sure you know how to read the pipette. You don't get a chance to do it a second time if you don't use the tool properly in the first place. So anyway, that diagram shows you what we're doing. This explains step by step what you're doing. This is how we dilute the first 6 tubes. This is where we put the bacteria on the Petri plate. And then we use the turntable and bent glass rod to spread them out. And then we incubate those 3 plates upside down in your Petri plate holder at 37 and place the used dilution tubes in the baskets in the hood. And that is the procedure for the plate count. So next time we'll do the plate count, put the results up on the board, and remember, do your practice problems in the online objectives for both the plate count and the direct microscopic count, so you can learn how to do those problems on the first lab quiz. And of course, any questions you have on that, bring to the results next time, and we'll go over it.